Our topic for today is regulation of gene expression. So, first, let us try and understand what is meant by gene expression. When we say gene expression, what is meant by gene expression? Gene expression means that gene controls or gene has all the information and it is used in the synthesis of a functional gene products. So what is happening? Gene is having all the information and this information is utilized to produce a functional gene product. And this product we know is normally the proteins. So the proteins are the end product of the gene expression. So a gene expression, we can define it as a process by which the information from a gene is used in the synthesis of a gene product, which normally is a protein over here. Now this allows the cell to respond to its changing environment. Now the unique feature of this gene expression is that it is both on and off switch. That means as per the cell's requirement, the gene expression, that means the synthesis of the protein, can take place or it stops. So it allows the cell to therefore adjust. Secondly, it also regulates the amount of protein synthesized. Regulates the amount synthesized. So here, this is the gene expression. Now when we are saying regulation of gene expression, what are the various ways by which, or what are the various steps at which the gene can be regulated? Now the regulation of the gene expression is different in prokaryotes as compared to that of eukaryotes. Now when we talk of the regulation of gene expression, we know in the case of let's say when we talk of the process of the expression of the gene, let us first try and understand how does it happen. This is the nucleus and this portion is the cytoplasm. The DNA carrying the introns and the exon, this is the DNA. By the process of transcription, mRNA is produced, that is the RNA is produced. This is the process of transcription. This over here is RNA splicing. And after the RNA splicing has been done, mRNA is formed and this mRNA is exported out into the cytoplasm, where the translation happens and proteins are produced. Now here you can see the process of formation of mRNA, that is the information for the particular gene product is carried out into the nucleus, and there it is then exported out where the translation or the protein synthesis take place within the cytoplasm. So in the case of eukaryotes, we can have a check or the regulation can be brought about at the transcription level first. Second, it can be brought about at the second level, that is the RNA splicing level, when the process of processing is taking place, the non-functional zones are removed. Also, during the transfer of RNA to the cytoplasm, that is from the transfer of RNA from the nucleus to the cytoplasm, and also at the translational level. So these four are the regulatory points in the case of eukaryotes. At each step, we can regulate the expression of the gene. Now when the in the case of when we talk of the prokaryotes, since they do not have well-defined nucleus or nuclear membrane, the transcription and the translation process both are taking simultaneously within the cytoplasm. So, in prokaryotes, the rate of the transcription, that is the rate of the transcription is can be controlled over there only at the transcription level. Now, at this point in the transcription unit, the activity of the RNA polymerase, that is always initiated by the promoter, can be regulated by the accessory proteins, or we call them as regulatory proteins. There are certain regulators, therefore, which can promote or which stop the expression of the gene. So we call them as operators. Operators, therefore, can regulate the gene expression, and these operators function in both ways. So we have operators, and these are the operator over there which promotes will be the regulatory operator, and the other will be the repressor operator or suppressor operator, which is normally the suppressor protein. So this is suppressor protein, this is promoter protein. This will promote and this will bind to the promoter site of the prokaryotic DNA and starts with the synthesis. So one is we will study about lac operon, which will have the promoter and the terminator so as to regulate the expression of the gene.